In our lab, we focus on the pathomechanisms of inflammation in patients suffering from inflammatory joint diseases, and more specifically, rheumatoid arthritis. In particular, we want to understand the role of cell-to-cell interactions as potential drivers of inflammation. We know that joint inflammation experienced by patients suffering from rheumatoid arthritis leads to pain, joint destruction, and ultimately joint disability.
In the recent years, different synovial pathotypes have been described in patients with rheumatoid arthritis, which are defined by the infiltration of different immune cells to the synovial tissue. While we are only just starting to understand the various cell types of the inflamed tissue by using single-cell-based technologies, the exact mechanisms and the factors that contribute to the development of synovial pathotypes remain unclear, which is precisely what we are currently investigating in our paper.
To investigate the interaction between immune cells and synovial fibroblasts, in short FLS, we generated co-cultures of human FLS and T cells. And in order to quantify cell-to-cell interactions, we established a bioinformatic image analysis pipeline for automated computation. From this data, we saw that cytokines, in particular TNF-alpha and interferon gamma, prime FLS to activate and differentiate T cells. And this is also in line with our transcriptomic analysis. Interestingly, we noticed that transcriptional signatures of FLS stimulated with pro-inflammatory cytokines were mostly associated with a subset of RA patients that have a so-called lymphoid pathotype. This subset of patients shows a high degree of immune cell infiltration in their joints. This is especially relevant in the clinic as these patients also show stronger symptoms and better respond to some biological DMARD therapies.
In summary, our work identified cytokine-activated FLS as some of the main conductors of synovial pathotypes. Together with our co-authors and collaboration partners, ongoing work aims to decipher the exact consequences of this cellular interaction and its role as a potential biomarker for treatment response and also for classification of rheumatoid arthritis patients.